Hi, my name is Sowon Kang, and I'm a rising senior at McNeil High School in Austin, Texas. And my STEM interests are biochemistry and biomedical engineering. So this summer, through the iSTEM program, I work with the Zhou lab at Boston College with Professor Jane and her undergraduate student, Catherine. So our lab mainly focused on engineering reverse transcriptase. And reverse transcriptase, also known as RTs, are enzymes that are present in retroviruses that go against the central dogma of biology by converting RNA to complementary DNA. And these RTs have many applications in the field of biotechnology. So the goal of our lab was to engineer an effective RT that can read through challenging RNA sequences. And our research has many real-world implications, as engineering RT that can read through challenging RNA sequences would help prevent many genetic disorders such as the Fragile X syndrome. So in order to achieve that goal, my role this summer in this lab was to compile a survey of mutated RTs with focus on thermostability, which is the ability of RT to function at a high temperature, and processivity, which is the ability of RT to continuously read through certain RNA strand and make DNA from that strand. And mainly, I focus on the RTs of human immunodeficiency virus type 1 or HIV-1. And this virus, HIV-1, consists of two strains, group M, which is the main strain, and strain N group O, which is the outlier group. And I focus on the outlier HIV-1 group O variant and study the mutations of group O variant and their impact on RT in terms of thermostability and processivity. And after I found these characteristics of the group O variant, I compared them with those of group MRT and other retroviruses such as the murine leukemia virus or MLV. So first, I started off my research by finding the sequence of the HIV-1 group O variant. And to do this, I read a scientific paper called The Characterization of the Reverse Transcriptase of a Human Immunodeficiency Virus Type 1 Group O isolate. And from this paper, the researchers obtained the purified nucleotide sequence of the Spanish isolate RT, known as the ESP1, compared the sequence with other HIV-1 sequences, and displayed their relationships in a phylogenetic tree diagram. So here in this diagram, you can see the area where I highlighted in a red circle, and that area, the HIV-1 group O variant, consists of the three nucleotide sequences, the ESP1, MVP5180, and AMT70. And after comparing these sequences, the researchers then derived the amino acid sequence of the RT coding region of the Spanish isolate and compared it with the sequences of other HIV-1 group O isolates. So in this table, you can see the sequences of the consensus O and the mutated sequences of ESP1, MVP5180, and AMT70. So from this table, I was able to obtain the amino acid sequence of the ESP1 and save the sequence via benchling. And then after I collected the amino acid sequence of ESP1, I compared the sequence with the sequence of the 1RTD, which is the sequence of the HIV-1 RT that our lab had already collected. And I compared these two sequences using the NCBI BLAST tool. So here you can see the query is the ESP1 and the subject is the lab RT. And this shows the area where those two sequences match and the areas where they differ. So these two sequences have 79% identity, and among their differences, we specifically focus on the mutation site K through 58R, A through 59G, and S368, which is the region that is highlighted in yellow. And we focus on this specific site because this was the mutation site that is known to affect the thermostability and processivity of the RT the most. So to analyze this mutation site, I found the crystal structure of the HIV-1 RT, and using the tool PyMol, I found the site of the mutation, those through 58, through 59, and 360. And these mutations were in the area that is highlighted in a red circle, and this was an interesting finding because these are the mutations that occurred near the loop structure of this crystal structure. After that, I looked at the chemical properties of these three amino acid mutations and analyzed the impact of these mutations in terms of thermostability and processivity. So out of these three mutations, the first mutation was K to R, lysine to arginine, and arginine can form more hydrogen bonds in lysine, resulting in an increased stability. And this mutation does not seem to impact the thermostability. And the second mutation was from alanine to glycine. And as you can see from the picture, the alanine and glycine are pretty similar in their structure and their properties. And the last mutation was serine to alanine, and alanine is missing the OH group of serine, so the replacement of serine by alanine was reported as a factor that increased the thermostability of the proteins. So this analysis would help how certain mutations of amino acids can impact the properties of RT in terms of thermostability and processability. And finally, I wrote a literature review over the scientific paper called Increased Thermostability and Fidelity of DNA Synthesis of Wild-Type and Mutant HIV-1 Group of Reverse Transcriptases. So in this paper, the researchers compared the HIV-1 group O wild-type RT, which is the one that is not mutated, with the RTs of MLV, HIV-1 group M subtype B, BH10 wild-type, and group O mutants, such as OV75I, OE478Q, and the combination of both mutations. And they have found that the HIV-1 O wild-type RT was more thermostable than the other RTs. So if you look at this bar graph on the left side, you can see that the O wild-type RT, which is the bar with the medium gray color, functions better than MLV and BH10 RT, even at high temperatures like above 54 degrees Celsius. And the researchers also found that even though OV75I and E478Q were less thermostable than the O wild type, the combination of both mutations was found to be more thermostable and maintain the accuracy of OV75I. So if you look at the graph on the right side, the mutation OV75I/E478Q is the one that is on lane 5. And you can see that it continues to function at high temperatures like 68 degrees Celsius, as it is highlighted in yellow color. And although it is not shown in this figure, the researchers also found that this mutation, OV75I/E478Q, was able to maintain the high accuracy of OV75I, which reveals itself as an enzyme with an excellent potential for future development. So after my work over the summer, since we now have a complete sequence of the HIV-1 group of variant RT and information about their thermostability and fidelity, we can use this information to engineer an effective reverse transcriptase that excels in thermostability and processability, which would be useful in reading through challenging RNA sequences and thus preventing various genetic disorders like the Fragile X syndrome that I mentioned in the beginning. And lastly, these are the papers that I referenced throughout my research, and that's it for my final symposium. So thank you so much for listening.